Good morning. Welcome to the Symposium La epidemiología genómica en las enfermedades infecciosas. The past 2 years represent a major challenge to our society, where science had a major role. All our technology and research were focused on the pandemic. However, we focus on one partner of this dance couple, the virus. In this scenario, the relevance of the host was diminished with the role of a fixed biological unit. In fact, on the contrary, there are subpopulations or even populations that can respond differently to the infection agents. Here, we want to bring up the host relevance, discussing different topics from the experience in animal and human health research. We are really proud and happy to have with us this morning 4 researchers that can give us different point of view of the same problem. All of them have a long-standing experience in their specific field, and I hope that they could forgive me for this brief presentation of each of you. Dr. Yáñez is an associate professor in the University of Chile. He rules the Aquaculture Genomic Lab, focused on selective breeding programs and the development of genomics research for the determination of disease resistance, growth, or product quality. The relevance of fish research compelled them to understand the disease susceptibility and create strategies and tools to deal with the problem. He combines the information from genome and transcriptome variability to understand susceptibility. Dr. Bonilla, on the other hand, is an assistant professor in the University of São Paulo. She has a long experience in study of the human genetic population, especially the mixed population and complex disease. She's working with the program ALSPAC, a cohort with more than 15,000 participants from the Bristol City area. It is the group that developed the Mendelian Randomization method. This method is like an influencer because nobody can ignore it, but for a good reason. It can be seen as a natural randomization experiment. Dr. Casanova is a Levy Family Professor at Howard Hughes Medical Institute, the Rockefeller University. He has a long-standing way of investigating the human genetic determinism of infection disease. His group is searching for what they define, and I really like this expression. They define as the monogenetic holes in the immune defense, and he has tens of publications of this topic. In the context of COVID-19, he rules the COVID Human Genetic Effort, an international consortium aiming to discover the relationship between human genetics and the different clinical presentation of SARS-CoV-2. And today, he's going to share with us some of the results. Dr. Doeschl-Wilson is the head of the Genetic and Genomics Division at Roslin Institute, University of Edinburgh. She investigates how the genetic of individuals affects the spread of infection disease by combining laboratory experiments with mathematical modeling and quantitative genetic theory. She will share with us methods basis, computational tools, and examples on super spreaders. At last, the aim of this symposium here is to try to go beyond the virus sequence and expand the vision on how to respond to a new infection disease. So we give all the screen to Dr. Yáñez.
Okay. Can you see my screen? Okay, so thank you, Bernardo, for the invitation. and for the nice introduction. So today I will talk about understanding host genomic variation for resistance or susceptibility to infectious and parasitic disease from a fish perspective. So in this presentation, I will try to summarize what has been our work in trying to dissect and to predict phenotypic phenotypes. Phenotypes from susceptibility or resistance in this in a group of vertebrates which are related with salmonids. So the main diseases which affect salmon and trout industry here in Chile are two. One infectious agent, Pichirrhiciasis salmonis, which is an intracellular bacteria that causes the also called salmon rickettsial syndrome, or SRS, and generates high mortalities in the sea growth period. On the other side, we have the Caligus royer-crucegi, which is a species of sea lice. It's an ectoparasite that causes injuries and decreased performance, and also facilitates the entrance of other pathogens. So the industry, or the aquaculture industry, treat these two agents using vaccines, antibiotics, antiparasitic drugs, and also some management measures. But all of them have not been as effective as we want in field conditions. So another option is to breed for less susceptible animals. So in fish, we have the possibility to perform experimental challenges using a big number of animals. For instance, we use from different families, like 200 families or 400 families from the breeding populations, using between 1,000 up to 3,000 animals. So all of them from different families. We use pit tags to keep the pedigree traceability. And for sea lice, we use infestation with between 50 to 100 parasites per fish. So as a trait definition or phenotype, we basically use the parasite load in the sessile stage on pectoral and caudal fins, or total count in the animal, to see if they are resistant or susceptible. In the case of the bacteria, Pichirrhiciasis salmonis, we use infection with a lethal dose 50 of inoculum to maximize the variance of the dichotomic trait, like dead or alive. But we can also use the survival or time to survival as trait definition. So the first objective is to see if there is genetic variation for these diseases. In the case of SRS, the bacteria, this is what you can see is basically we have the families in the x-axis and mortality, percentage of mortality in y-axis, and you can see a kind of same pattern in three different species. Atlantic salmon, rainbow trout, and coho salmon. So there are different range of mortality rates across the different families. Each bar here in this bar plots is a different family. So there is considerable between family variation. This is the case of the Kaplan-Meier survival curves. So you can see variation across time with, between different time points. And these are, for instance, the most resistant family and the most susceptible family. So something very important to know if we can breed animals to be more resistant to this bacteria is to estimate genetic parameters. So we need to know the heritability of the trait, And here you can find, or you can see that the heritabilities for the bacteria resistance in different, in the 3 different species: Atlantic salmon 0.25, rainbow trout 0.44, and coho salmon 0.16. So there is significant genetic variation for resistance or susceptibility to the infection of this bacteria in these 3 species. So this means that we can select for animals which are more resistant to the bacteria. In the same manner, if we go to see what happens with the parasite load with lice count across the different families in experimental challenges, we can see that in Atlantic salmon and rainbow trout, there is the same pattern of phenotypic variation across families, and heritabilities are ranging or are kind of low, 0.1 and 0.11, but still significant. So we can also have some room to select for animals that are more resistant to the parasite. Okay, so these are large-scale experiments that allowed us to understand or to identify genetic variation for bacterial and also sea lice resistant. So in terms of selection, we need to predict the phenotypes, right? If we use only pedigree, that means if we use only family-based selection, the estimation of breeding values is not that accurate. So we have low accuracies when predicting or estimating, predicting the phenotypes or the phenotypes of the progeny of the animals when we use only pedigree information. And also there is a conflict or trade-off between selection intensity that we can apply and also inbreeding, because we will tend to select animals from the same families. So the consequences of this are that we have a limited genetic progress. So if we can incorporate, or if we can integrate pedigree data, phenotypes, and also SNP markers information, estimating the markers effects across the different or several thousands of SNPs, we could try to generate genomic predictions, right? Which will result in theoretically in increased accuracies of the prediction of the genetic merit of the animal, increase the response to selection, and accelerate the genetic progress. So how does genomic predictions work? It's basically that we have a training population or a reference population in which we know the genotypes and the phenotypes of the animals. So based on these animals that have genotypes and phenotypes, we fit some prediction equations, and then we predict the genetic merit of selection candidates that doesn't have phenotypes, only with marker information. So we use sparse markers, uniformly distributed markers to predict the genetic merits of selection candidates based on information from genotypes and phenotypes from the animals. So that's why we make selection decisions, and we use what is called genomic breeding values, which is similar to polygenic risk scores in humans. So they're kind of models. So one thing that is important in animal breeding is how we can maximize the phenotypic prediction. So if we use only phenotypes and pedigree, these are the range of accuracies in terms of predicting the phenotypes that we can get. So this is the accuracies. This is basically the correlation between the true genetic merit, the true breeding value, and the estimated breeding value. So this is using pedigree blab, best liner and base predictor. If we use an HD array and high density SNP array, we are here, around here, in this distribution denoted by the blue line. So we increase the accuracy if we use SNPs. But if we use whole genome sequences, the accuracies are not that different to those presented using an HD or high density SNP array. So One of the hypotheses was that if we have the sequences, we have the full information related to the genetic variants affecting one trait, but it seems to be that sequences also provide some noise. So accuracies are not greater than or higher than using an HD SNP array. But if we use the QTLs or QTNs, quantitative trait nucleotides or quantitative trait loci, that means the causative mutations that are affecting a particular trait, we can maximize the prediction accuracy. So these are simulation studies present in this paper from Miguel Pérez Enciso, and it's very important to see that we have to aim to get information from functional variants. This is something that we have tested in salmon, Atlantic salmon, rainbow trout, and coho salmon. So we basically saw that, in fact, when we go from pedigree to high density SNP markers, from the black line to the blue line, we actually tested this empirically, and we saw these differences in accuracy. These are the accuracies for genomic prediction for the bacterial resistance in the 3 different species, comparing pedigree blab only using pedigree and phenotypes information, and in orange here, compared against using genomic information, different methods. We tested different methods. Is related to the blue, light blue bars here. We use frequentist approaches, GBLAB, and also some Bayesian approaches, Bayesian lasso, Bayes-C, in the different species, and we always see an increase in accuracy of genomic predictions when using SNP information. Okay, but as you can see, these accuracies are kind of different to unity, so they are not maximum. We saw the same for the sea lice. Here we tested different marker densities from 0.K up to 50K, different methods, and this is relative to pedigree blab. So we always get between 10% of increase in accuracy up to more than 20% of increase in accuracy when we use SNP information. In terms of the genetic architecture of these traits, they are both of them are highly polygenic. In terms of SRS resistance, we tested a Bayesian genome-wide association approach to test binary survival and day-to-day in the different species. And then we used comparative analysis or comparative approach between the 3 species to identify the most important putative genes or candidate genes to be related to the disease or to the susceptibility of the disease. So as you can see, there are a lot of markers that that explain a high proportion of the genetic variance of the trait, but they have low statistical support. So and this is basically due to that the effect of the SNPs is not so high. This is the list of the list of genes that we found when comparing the 3 different species. So some of these genes have been explored by different groups. For instance, NLRC3, which is right here. And a different group tested this particular gene, which has been associated with the response of the host to another intracellular bacteria, basically in humans. And it seems to be that this gene is important, and with some functional information, there is already confirmation that some of these genes are important for the susceptibility of the trait, so of the disease. So as you can see, there is no one particular gene. There are different genes, Some of them are not that obvious, involved in the immune response. We can say that the response or susceptibility to bacteria is polygenic and complex in terms of the architecture. Going to the genomic architecture of sea lice resistance, we perform a genome-wide association study, and we only found one gene surpassing the significance threshold when we used a couple of thousands of animals and 50K SNP array. This gene was related to collagen A1, which is involved in the repairment of injuries in the skin. So it's an important candidate. But so we face here polygenic traits, and when we face susceptibility, or we try to understand susceptibility to bacterial and parasitic disease, and it's kind of different story when we try to understand the genetic architecture of susceptibility to virus. There are a lot of examples in aquaculture or in different vertebrates that there in virus, the response or the genetic variation for susceptibility is controlled by few loci or few genes. But here where we are facing polygenic traits. Bacterial and parasitic susceptibility are polygenic traits. So how to increase statistical power and accuracy for detection of QTNs when we face polygenic traits? One option is to use a combined approach between imputation to whole genome sequences and meta-analysis. So we tested this and we make an effort together with a company called Aquagen, which is the main genetic provider for salmon and trout. In terms of Atlantic salmon, we used 10,000 animals, genotype with 50K SNP array, more than 1,000 animals, genotype with 200K, and 1.3 thousand animals, genotype with 600K SNPs, and also almost 300 fish that were sequenced using whole genome sequence approach. So this is the total of animals used in the next steps. We went from 50K to 200K. These are the SNPs that we imputed with accuracy higher than 0.8, 98%. Then we went from 200K to 60K, and then to 60K from whole genome sequences, reaching 2.4 million SNPs that were imputed with accuracies higher than 0.8. So with this high resolution information, so we went from 50K to 2 million SNPs. We perform meta-analysis for disease-resistant traits for SRS and sea lice. So this is the accuracy of imputation from 50K to 200K using, in each chromosome, just a picture of that. And these are the number of SNPs at different levels of imputation accuracy from 600K to whole genome sequence. So we were very sure that the SNPs that we used were imputed with high accuracy, and we exploited information from these imputed whole genome sequences to identify potential QTNs using meta-analysis. This is the case of the bacteria. So we basically had 4 populations with different. This is the number of animals on each population. This is the number of SNPs that were used in the beginning prior imputation, and then these are the different conditions of the challenge test, the mortality, final mortality, and duration of the experiments. So we found some QTLs or some regions that were important for explaining the susceptibility of the trait, and they were mainly related with the genes that we provided in the table previously shown. So something that was very important in this analysis was that from the side of sea lice, we were able to find a lot of new genes. So for SRS, we rediscover or we confirm that the genes that we found in the 3 different species were important in controlling the trait. But here we, for sea lice, using 6,680 fish with phenotypes and genotypes, imputed from 50K to 500,000 SNPs with high accuracy of imputation, and 3 different traits, lice count, lice density, and log lice density, we discovered new genes, right? Some of them are very obvious or important candidates, like GMAP8 or FGFR, and also we confirm, for instance, collagen, but still this is a highly polygenic trait. So what are the future directions? We are trying to prioritize SNPs to perform genomic predictions, as is in this case. This is not related with disease resistance, but is related with thermal stress. So there is significant genetic variation for thermal stress in rainbow trout. We performed GWAS using imputed genotypes, and we selected SNPs to based on GWAS hits to increase the accuracy of genomic predictions. These are the main results. When we use pedigree blood, this is the mean accuracy, 0.66. This is the accuracy using 50K randomly selected SNPs. When we use whole genome sequences, the accuracy doesn't increase a lot. When we used 22K SNPs selected or prioritized based on GWAS hit, we reach a maximum accuracy or higher accuracy than using previous method. So functional studies are very important. This is an international initiative, the functional annotation of all salmoniid genomes. We are working on this initiative, trying to get more information on functional information related to causative mutations involved in disease resistance. And these are the conclusions. SNP panels have helped in uncovering the genetic basis of disease-resistant traits in salmon and trout, both polygenic and monogenic or oligogenic traits. In this case, bacterial resistance and parasitic resistance are highly polygenic traits, as in difference to viral diseases, which are mainly oligogenic traits, or resistance to viral diseases, which are mainly oligogenic traits. Genomic predictions increase the accuracy of phenotypic prediction for disease resistant in salmon and trout, and meta-analysis of GWAS using ultra high density, that is SNPs from whole genome sequencing, boosted the statistical power to detect genetic associations for polygenic traits. And when we use SNP information from prioritized variants, we can increase the genomic prediction accuracy by means of selecting these SNPs and using them into the genomic evaluation models. And also functional studies will be key to increase the rate of genetic gain and also the accuracy of genomic predictions in the near future. This is an invitation to attend to the International Symposium of Genetics in Aquaculture that will be held in Puerto Varas in November and December 2022. And thank you all for your attention.
Well, thank you, Dr. Yáñez, for the presentation. The questions are going to be at the last moment of the symposium. And so at this moment, we want to give the talk to Carolina Bonilla.
Thank you, Bernardo.
Okay, I will share my screen now. Can you see?
Please, Carolina, you need to expand.
It's expanded.
In the other way that you used it previously.
Yeah, it has to be like that.
All right, that's okay.
That's okay. Yeah, this way I won't be able to have the pointer though. Maybe you can see my mouse there or not? The movement of the mouse?
Yeah.
Okay, so to begin, I would like to thank Bernardo and the ALAG 2021 Organizing Committee for the kind invitation. I'm really happy to be presenting at this symposium and be sharing the stage with all the other speakers. And I will be telling you about something very different from the previous talk. I will be talking to you about a relatively new method called Mendelian randomization and how that can be used to investigate host factors for COVID-19 susceptibility and severity, and in general, also in other infectious diseases. So the outline of the talk is as follows. Do you see my next slide? Are you seeing my slide of the outline of the talk?
Yep.
Okay. So sorry, I was confused. So the outline of the talk would be as follows. First, I will explain a little bit about the rationale for using Mendelian randomization. I will describe some of the features of Mendelian randomization. Then I will go on to talk about some examples of MR applied to COVID-19 disease. And then I will end with a summary and some recommendations. So about the rationale for using Mendelian randomization, it comes from the shortcomings that can be seen in observational epidemiological studies. I know that most of us realize that observational studies in epidemiology are susceptible to a number of biases, such as selection bias, memory bias, information bias, confounding, which is very important, and also reverse causation, which is the case when the outcome influences exposure and not the other way around. So it seems that every day there's something coming up that some exposure is causing some kind of disease in a different group of people. So because observational studies are subjected to all these problematic issues, then their results appear inconsistent. So what can we do about that? Some of the possible solutions in epidemiology are within also observational studies. We could do a cohort study where you recruit individuals to your study based on the exposure or the exposure phenotype, whether they have been exposed or not exposed to a particular risk factor. These individuals are then followed up in time, and after that time, you register whether they have developed the outcome of interest, usually a disease, and then you compare both groups, the exposed and the unexposed. This is a good design in terms of that you can see that the exposure occurred before the outcome. And that allows you and provides you a lot of evidence to establish causality, because epidemiology wants to find causes for disease. However, the cohort studies still have some problems, especially loss to follow-up. Individuals get tired, fed up, they leave the study, they move cities, they die for other reasons. And it's also an expensive design because you have to have funds and logistic and the energy to put together a large number of people and follow them across time. Another possible solution is to run a randomized control trial. This is not an observational study, this is an experimental study where the researcher will intervene and manipulate the exposure. This type of study, what it does is collects a group of individuals, recruits a group of individuals to the study, and these are randomly allocated to different groups in which you are going to test an intervention, be it a new therapy, a new treatment, and one of the groups will receive that treatment or that new therapy, and the other group will receive the standard of care or a placebo. These kind of studies are also very expensive and sometimes are not ethical. So, for instance, you can't randomize people to smoke or to drink alcohol. So, based on all that, some people started thinking about another option, another avenue to try to identify causal relationships in epidemiology. And these two epidemiologists here, George David Smith and Shai Ibrahim, which were at Bristol at the time, George is still there. They published this paper in the International Journal of Epidemiology in 2003, where they described what they call this new method that they called Mendelian randomization. The idea for Mendelian randomization had been there before, but they were the first to instrumentalize it. So Mendelian randomization is about using genetic variants as proxies for modifiable exposures to establish causal relationships between these exposures and an outcome of interest. So what is the basis for Mendelian randomization and what does it get that name? First of all, it is based on Mendel's laws, the principle of segregation, which has some random element there, and the law of independent assortment. And aside from that, it is also based on instrumental variable theory, which comes from econometrics. So instrumental variables, let's say that you have a model of interest that you want to find out whether some exposure X1 is associated with an outcome Y, and this relationship can be biased by the presence of another variable called X2, which could be a confounder, for instance. An instrumental variable is a variable that does not belong to this model, to this equation, but it's strongly associated with the exposure, and importantly, is not associated with the error term. So it will allow, using the instrumental variable, that will allow you to obtain more consistent and less biased estimates. Mendelian randomization, therefore, has three main assumptions. First of all, is that the genetic variant, which will be the instrumental variable in this case, is associated with the exposure of interest, and that is the principle of relevance. The genetic variant is not associated with the confounders of the relationship between exposure and outcome, and that is the principle of independence. And finally, the genetic variant is associated with the outcome only via the exposure, and it is called the exclusion restriction principle. So here, if you see this diagram here, you have an exposure X and an outcome Y, and we want to find out about this relationship. There are confounders associated with both of them, and then we use this genetic variant that is associated with X to estimate this effect. The dotted lines represent the coefficients that we have to calculate. We have to calculate the coefficient of association of the genetic variant with the exposure, which is beta sub X, and the coefficient of the genetic variant, the association of the genetic variant with the outcome, which is beta sub Y. And those, simply, you divide those to find the causal effect. So it's beta sub Y over beta sub X, and this is called the Wald estimator to find out the causal effect of the exposure on the outcome. Mendelian randomization is becoming more and more popular, and after the publication of that paper, it really picked up. And this is due mostly to a few things. No? So, for instance, the availability of GWASs, the fact that GWASs are easily conducted nowadays with lots of people, And the technology has become cheaper as well, and it has allowed us to have all this wealth of data to see associations of SNPs, not only with the typical phenotypes, but also with methylation and transcription. And in addition to that, which was really important, and I really want to make an emphasis on this, that data was able to be shared. And there were data repository, and there are the publicly available data that everybody can use. So Mendelian randomization has been compared to a randomized controlled trial, and I'm going to show you here how that works. So in a random, if you look on the right-hand side, in a randomized controlled trial, you will have a population where you want to carry out that trial. And let's say that we are testing LDL cholesterol lowering therapy. This treatment is randomly allocated to people, and these people are going to end up in one of two groups. One is the treatment arm, and the other is the usual care arm or the placebo arm. Because of that randomization, which is actually the most important feature, individuals are going to be, the confounders are going to be evenly distributed between these two groups. The only difference that you expect to have between these two groups is the treatment. Then you follow up the individuals and register the incident major cardiovascular events that occur in each of these groups. So if there is a difference between the number of events in each group, then that can be assigned to the difference in LDL cholesterol that was provided or caused by the therapy. In Mendelian randomization, there's something similar. Only that your treatment is the allele that you receive from your parents. So where there's a natural allocation, random allocation of alleles happens in the population for a particular SNP that's associated with levels of LDL cholesterol, and this was identified or detected in a GWAS earlier. And so some people in the population will receive the allele that lowers LDL cholesterol, and that will be equivalent to the treatment arm. And other people in the population will receive the other allele, and that will be equivalent to the usual care arm or the placebo arm. Again, because of that randomization and because of the independent assortment, you expect the confounders or the other traits to be evenly distributed between these two groups. And if you then found differences in major cardiovascular events between these groups, you could think that that is due to the difference in LDL cholesterol level that was given by the different genotype. So in epidemiology, a randomized controlled trial is considered the golden standard, and Mendelian randomization has been called nature's randomized trial for that reason. So I'm going to show you now how Mendelian randomization can track or can foresee results of randomized controlled trials. So this was a work that we did when I was still at Bristol about 4 years ago. And we wanted to see whether circulating selenium levels were associated with prostate cancer risk. Particularly, we wanted to replicate what was seen by this randomized controlled trial called SELECT. This was a large trial that involved several thousand people, and where they allocated selenium supplementation to one of the groups in the trial, and the other group received placebo. There was some evidence before that increasing selenium levels could protect against prostate cancer. So this trial, they allocated individuals to the groups, and then eventually, at some point, they decided to look at whether there was any evidence for that relationship, for that protective relationship, and they did not find anything there. After that, and that trial was suspended. After that, we decided to look at whether we could see if Mendelian randomization could have foreseen that. So we used SNPs that were strongly associated with selenium levels in blood, and we had the exposed group, which were individuals who had the higher selenium alleles, and the control groups had the other alleles. And we compared the rates of prostate cancer between those two groups, and we again found nothing there. So it was very similar. We obtained very similar results, but the funny thing is that this SELECT trial not only did not see the expected relationship between selenium and prostate cancer, but also they they detected some side effects. So people who had prostate cancer were having a more advanced prostate cancer, and they saw an increased risk for type 2 diabetes. So this select trial was stopped after it had already costed $114 million. So you can see that Mendelian randomization is a way that you can maybe test beforehand, before embarking on carrying out these really expensive trials, to see whether there is some evidence of that association. Mendelian randomization, there are two big ways of doing Mendelian randomization. One way is to do one sample, Mendelian randomization in one sample, and that means that you are going to have to genotype everybody in your sample and measure the exposure and the outcome also to all those people. This uses individual level data, so you will have data for each individual, and it is very likely that you also have data on typical confounders. Mendelian randomization in general requires large sample sizes, and in this situation of the one sample, these sample sizes are hard to obtain because, let's say, if your exposure is easily measured, like BMI, it's not that problematic. But if your exposure is, let's say, vitamin D levels, then that requires lab tests, and that makes it very expensive. And if you have to type a large number of people for that, cost will really increase. The benefit of doing one sample MR is that it allows you to test for assumption number 2, which was that the genetic variants were not associated with the confounders. You can do that in this kind of setting. However, the type of Mendelian randomization that is mostly done nowadays, because it's easier and the data is there and it has more power, is two-sample Mendelian randomization. And in this case, what you use, instead of using one population, you use two samples. So the genetic instrument exposure association will come from one sample, let's say from one GWAS, and the genetic instrument outcome association will come from another sample. In this case, you are using summarized data, which has the advantage that they are easy to share, and you don't have to ask for the ethical permissions that are required to use individual level data. The samples have to be independent, but they have to belong to the same origin population. Let's say you have to use samples that are both Europeans, or no, it's not worth doing it. One sample from Chinese GWAS and another from an African GWAS. It's not going to work like that. However, because you don't have individual level data, you're not going to be able to test the association with confounders. But this type of Mendelian randomization has a lot of statistical power because of GWAS nowadays. They have a large numbers of individuals, and they can estimate precise effects. And so Mendelian randomization is not a panacea. It has some good features, like the ability to establish causality, because the genotype always occurs before the disease. So that temporality factor is there, that it's unlikely to be confounded, and also it will not be affected by reverse causation because of that thing as well, of the fact that the genotype occurs at conception and always before any appearance of the disease. However, it has some limitations. The main ones are pleiotropy, so genetic variants are likely to have more than one effect, and if they do have direct effects on the outcome, or an effect on some other factor that then has a direct effect on the outcome, and that doesn't go via the exposure, then it poses a problem. It can bias your results. However, there have been methods development that tackle that problem. And then population stratification can still confound the results because it can be a confounder of the genetics associations. So going now into COVID itself. So I did a literature search and identified about 60 studies that have looked at several risk factors, a variety of risk factors for COVID outcomes. And the outcomes that were used were infection, severity, and death. And there were all of these different risk factors for COVID tested, and all of them, of course, had SNPs associated with them. So that why was Mendelian randomization possible to be carried out. In red, I am showing here two factors, smoking and vitamin D, and because I will talk a bit more about them in the next slides. On the other hand, you could also use COVID susceptibility as an exposure and test its association with other outcomes, and that was done in fewer studies, which is understandable because initially you want to know what the risk factors are. So these are the outcomes that were tested against COVID susceptibility. The COVID susceptibility needs instruments for COVID, and these instruments were obtained from a GWAS that was carried out by the Host Genetics Initiative with hospitalized individuals and individuals who reported a COVID infection, and they have found some SNPs associated with that can be used as instruments. So going straight into smoking and COVID, this is a paper by Clift and colleagues, and they, as outcomes, they assessed SARS-CoV-2 infection, hospitalization, and death. And the exposures that they look at were smoking initiation and heaviness, which is how much you smoke. The instruments that were used were for smoking initiation, 387 SNPs, for heaviness, 57 SNPs. And these were obtained from the GScan consortium that included 25 GWASs. The population was European and white British. The type of the study was a case control study, and the strategy that I used, so they did, on the one hand, an observational study, and then on the other hand, a two-sample Mendelian randomization study where the smoking, the SNP smoking association came from the GScan, and the SNP COVID association came from the UK Biobank. So here are the results that they found. So these are the observational results. So they ran two studies, one age and sex adjusted, two models, sorry, age and sex adjusted, and another that was maximally adjusted for lots of other confounders. And so on top you have infection, hospitalization, and death in that order. And if you look at these forest plots on the right, you can see that former smokers were at an increased risk of developing COVID infection. In terms of hospitalization, former smokers and current smokers were also at an increased risk of being hospitalized. And in terms of mortality, it is very clear that there's a higher chance of dying for former smokers and current smokers, and within current smokers, those who smoke more than 10 cigarettes a day. So this is their observational results. Going to the Mendelian randomization results, they found something similar. For smoking initiation, individuals who were susceptible to start smoking, they were at higher risk of developing a COVID infection and hospitalization. Death, there was an indication that could be an increased risk, but the association was not, the evidence was not strong. And with smoking heaviness, the more people smoke, the higher the risk for being infected with COVID, the higher risk for being hospitalized, and a really high risk of dying. So this is an example where Mendelian randomization and observational epidemiological studies agree. And this has been replicated sometimes, some additional times, and this is now, I think, fairly established. Going into vitamin D and COVID, there wasn't a paper that combined both things, so I extracted information from 3 different papers. This is a paper that looked at outcomes, COVID infection severity and mortality. The exposure was 25 OHD status, which is the circulating vitamin D. And they did a systematic review and meta-analysis of 39 studies. The studies were carried out in worldwide in a number of different populations, although the majority were in Europe. The results for this study, I only, in this slide, I only put severity and mortality, but infection or susceptibility was very similar. However, in this case, the authors are showing vitamin D deficient. So the exposure they are showing is vitamin D deficiency. So in this forest plot, for instance, for severity, you can see that individuals who are vitamin D deficient are at a higher risk of having severe COVID. Similarly with mortality, individuals who are vitamin D deficient compared to those who are vitamin D sufficient, they are at a higher risk of dying. So that's their conclusion. Seems to be a positive association between vitamin D deficiency and COVID outcomes. Going into the Mendelian randomization results, there were two papers that reported very similar results. They looked at SARS-CoV susceptibility, severity, and hospitalization. The exposure was again 25 OG levels. And as instruments, one of the study used 112 SNPs and the other used 80 SNPs. The population were European. The type of study was a case control, and the strategy was a two-sample MR study. And the SNP vitamin D association came from the UK Biobank, and the SNP COVID association came from the Host Genetics Initiative. The results were very similar, so I'm showing the results from just one of the papers. If you focus on the top panel, you can see that the three different outcomes, and the main Mendelian randomization method, which is on the first line, is inverse variance weighted, and the others are methods that are used as sensitivity analysis to test if they are violation of the assumptions. And you can see that the effects are bang on the odds ratio 1 line. So there's nothing there. There's no effect detected in these studies. And then finally, I want to show you a result for a randomized controlled trial that has been carried out. There were only 2 trials carried out with vitamin D supplementation. This one is the bigger one, and it was carried in about 200 people in Brazil. And what the authors did was have a group that was supplemented with a large doses of vitamin D, and the other group received placebo. And the outcome that they measured was hospital length stay in COVID patients. So on the first, on the left-hand side graph, they report the hospital length of stay in the x-axis and the probability of hospital discharge on the y-axis. And these are the curves in black for the vitamin D supplemented people and in orange for the placebo people. And you can see that there's really those almost overlapping curves. And they tested that there was not a significant difference. When they, and this was considering all patients, when they considered patients who had arrived or entered the study with a vitamin D deficiency, again, they saw no difference between the supplemented individuals and the placebo individuals. So this is to show that at the moment, the evidence from an RCT and from Mendelian randomization does not support the use of vitamin D supplementation in the population to prevent COVID or to prevent severe outcomes of COVID. So at the moment, we do not have enough evidence to do that. We do have some evidence from observational studies, but because they suffer from more biases, their evidence has less strength compared to an RCT or a Mendelian randomization. So in summary, I would like to say that Mendelian randomization is one of several genetically informed methods that can be used, and they are powerful tools to help elucidate the causal effect of exposure on disease or exposure risk factors on disease outcomes. There are statistical approaches that can help dealing with Mendelian randomization limitations, in particular with the violation of the main assumptions. Mendelian randomization is an area that's very, very hot, let's say, and there has been a lot of methods development. There are several extensions that can be used in different situations. But we have to recognize that MR studies can be complex and interpretation can be challenging. So it's not recommended to just use Mendelian randomization. It's always the findings should be considered alongside other sources of evidence, and that is called a triangulation of evidence. And finally, I would like to just briefly run through what does it mean for us Latin Americans. Can we do Mendelian randomization? Mendelian randomization requires large sample sizes, so there's some things we can do. At least we can start doing. And a student of mine, Lara Baccarini, and I published this paper last year in Genes, where we have some suggestions for how we could strengthen the evidence in order to find causes for chronic diseases in Latin America. And this is it for me, and I would like to thank you again for your attention.
Thank you very much, Carolina, for the talk. It's really interesting, this method. So now I will the opportunity to present the research to the Dr. Casanova.
Well, thank you for inviting me.
Thank you very much, Dr. Casanova. Really an enlightening talk. The questions are going to be at the last of the symposium.
At what time? Because I have other Zooms later. At what time will the questions be?
In half an hour.
All right, good. See you then.
Yeah. Okay. So now we give all the screen to Dr. Andrea.
Okay. I hope everybody can see my screen and can hear me okay.
Yeah.
Hello, so my name is Andrea Doeschl-Wilson from the Roslin Institute, and many thanks for inviting me to give a talk. Right, so I work in a livestock institute, so most of us work on livestock, and I lead a research group there, and one of the key questions we try to address is what role does the host genetics play in the spread of infectious diseases? And for livestock, we can be braver and ask the next question, well, how can we use what we learn about the genetics to actually prevent outbreaks that we see in these pictures? So what is the status quo? Jose already described it a bit in fish. So there is now overwhelming evidence that there's a lot of genetic variation between individuals in their resistance to disease. But to be honest, we struggle a bit in identifying the genetic signal in the disease data we see, because there's so much noise in these data. And because of that, breeding for disease resistance is actually quite limited. So we have this amazing tool in livestock that we can breed animals with greater resistance and hence reduce the spread of the infections or the prevalence of disease in a population. But in reality, we struggle. And I showed this paper here at the bottom because that was one of the key recommendations of my colleagues, Steve Bishop, the late Steve Bishop, and John Williams, saying we need to get better. And in order to get better in dealing with disease data in the quantitative genetics scheme is that we need to implement epidemiological principles in our genetic models. And indeed, when you compare the quantitative genetics approach to disease and the epidemiological approach, there is fundamental differences, and I want to talk you through that. So in the quantitative genetics approach, the way we handle that situation is a bit what we see here. So we assume that animals or individuals are equally exposed to infectious pathogens if they are in the same herd. And some individuals may be more resistant genetically to becoming infected than others. In reality, most studies that look at disease resistance or the genetics of disease resistance, actually we find that the resistance is more in the way that animals cope with the infection. So rather, very few animals are actually resistant to becoming infected per se, but there's a lot of variation in the ability of coping once you are infected. And what matters here is that infected individuals, they can also, they have pathogens, they shed pathogens, and they can transmit pathogens to other individuals. But in quantitative genetic studies, we usually ignore the genetic effects on the transmission. We look at genetics for resistance, but we don't look at the genetics underlying the transmission. In epidemiology, however, the focus is completely on transmission usually. So you have two host traits that affect the transmission of infectious particles. One is the susceptibility to disease. So that's the propensity of an individual to become infected when it's exposed to infectious pathogens. And the other host trait is the infectivity of individuals. So that's the propensity to transmit the infection to susceptible individuals given infection. But in contrast to geneticists, epidemiologists hate variation usually. So they usually ignore the genetic variation. And that's a bit of a shame, because there's a lot of evidence that there's a lot of individual variation in infectivity, in the ability to transmit infection. And indeed, especially also for COVID-19, you will have heard probably the term super spreader quite a lot. So a super spreader super spreaders are individuals, or the small proportion of individuals in a population that contribute to the majority of transmission events. And previous studies have found that many of the large-scale epidemics or pandemics usually are connected with super spreaders. So they play a fundamental role in disease outbreaks. Now the question is, to what extent is super spreading or infectivity genetically controlled? We really don't know this at all at the moment. But just pause for a moment to think, imagine we could identify a potential super spreader based on its genetic material a priori. That would mean we could prevent a lot of pandemics and epidemics to happen. So the following questions really arise. If there was genetic variation in infectivity, can we detect it? What kind of data or models are required to see it? So you could, for example, imagine that individuals vary genetically in their shedding. So they shed a different amount of pathogens. And because of that, they vary in their ability to transmit. But unfortunately, pathogen shedding is not a trait that you can easily measure. Can we do better? Can we infer infectivity from data that we have? And once if we can detect genetic variation in infectivity, the next question comes, how big is it? How much genetic variation is there? And more important, how is it correlated with resistance? Are the animals that we want to breed for increased disease resistance, are these the potential super spreaders? Could well be, because they cope well with the infections. Maybe they are the ones that spread a lot. And that brings us to the next question. Should we include infectivity as a new trait into our genetic selection programs in livestock? Should we breed not only for increased disease resistance, but also for reduced infectivity? So we don't know any answers to these questions at the moment. And the way we approach this in our group is threefold, step by step, very systematically. So the first thing is we realized we first tested existing genetic models to see, could we detect genetic variation in infectivity if it was there? And we found that the existing quantitative genetics machinery doesn't do very well. So we started to develop our own methodology and validated our methods on simulated data. So that was the first step. So we used a lot of simulation models where we simulated epidemics just to see what does it take to actually detect genetic variation in infectivity. Then the next step is, then we conducted disease transmission experiments. And that was in different species, and I show you some examples today, where we have experimental data to estimate genetic effects for susceptibility and infectivity. So the two driving traits that drive the disease spread in a population. And of course, you can see the benefit of experiments over field data is that they are much less noisy. So it's much easier to detect the genetic signal. But eventually we need to venture into field data because we need a lot of data, like the Mendelian randomization, we need a lot of data to estimate infectivity. So we need to go to the field data, but that's the next step. So I just want to talk you through these 3 steps a little bit. So first to the methods, that actually is still work in progress. It involved several PhD students and postdocs, but we are getting there. So the latest version of our software for estimating susceptibility and infectivity effects is in GitHub there. And there are some published papers and more are just to come soon. But this is roughly how the methods work. So we have this software, it's called SIRE, which stands for Susceptibility, Infectivity, and Recoverability Estimator. And as input data for the software, what we need is, first of all, individual data of individuals' infection or survival status. So often we don't know if an individual is infected or not, especially in fish, what José showed before. Very often we only know if a fish is alive or dead, or an animal is alive or dead. But we need individual data of animals over different time periods. And we also need pedigree or genotypic data, then of course for the genetic models. The software uses this data to estimate either breeding values or genetic risk effects, polygenic genetic risk effects for susceptibility and infectivity, or SNP effects, and also the true infection times of the individuals, because usually you never know when they're truly infected. And the way it does it is by combining 3 types of models. So you have first a classical genetic animal model that links the genotypes or the breeding values to the phenotype. So these are susceptibility and infectivity values. The second type of model that this software incorporates is an epidemiological model that links the susceptibility and infectivity of individuals to their true infection times when there is infectious pathogens in the populations. And the third type of model is an observational model that links the unobservable true infection times to the observations. What do we actually see of the infection status of this individual? I realize this is a very short description, so please do go to the papers if you're interested or ask questions. But with this software, what are the key findings to date when we apply them to simulated data? So first we find actually, yes, it is possible to get accurate and unbiased estimates for genetic infectivity, as well as susceptibility or recoverability, if we have the appropriate data. And so if there were genetic superspreaders in the population and you have the right data, you could see them. Now, the right data doesn't mean we need new measurements. We can actually estimate them from the measurements that we often have readily available, such as the diagnostic test results. Is an individual infected or test positive for infection or not at given times? And often survival is enough, which I will show you later. However, what we do need is slightly different samplings or field study designs. And in particular, we need to have not just data on many individuals, but we need to have data on many epidemics, so different herds that are exposed to the same, to infectious pathogens. And we need to have temporal information of individuals' infection status. But the good news is that we can actually get robust estimates even when our data are noisy or incomplete. So you do not need to have perfect data to estimate infectivity. So I want to talk you through a few transmission experiments, what we found. With the methodology in place, we are ready to do some experiments. And so we have done it on several species, but today I just focus on Atlantic salmon for 2 virus infection in Atlantic salmon. So the first one is what I call the IPN lucky case. It's infectious pancreatitis necrosis virus in Atlantic salmon. which causes high mortality at the freshwater stage and as well at sea. And what is this is a very exceptional case because in this case, a single QTL explains most of the genetic variation in mortality of the fish. And of course, the breeding company have taken this on board and have started to breed for disease resistance based on this QTL, so to marker-assisted selection. And actually in the future, that will probably soon replaced by gene editing. And as you can see in the graph on the right here, mortalities have drastically reduced due to the selection. So genetic selection, breeding for disease resistance really works. Now, we wanted to see, does actually this QTL infer any differences in susceptibility? What is the effect of this QTL on transmission? Are the so-called resistant type of fish, so the fish that carry this R allele, the beneficial resistance allele, are they less susceptible to becoming infected? And if they're infected, are they less infectious? So together with FIMA in Norway, we conducted quite a small-scale transmission experiment, actually. And in this simple experiment, was very simple. We had infected shedderfish that we inoculated with the virus, and then we put them into tanks with susceptible fish that were not infected to spread the infection. And we distributed the fish according to their resistance genotype. So here, the fish that have an R, they carry the beneficial resistance allele. So what we did is we monitored in every tank. We recorded the time to death. So we only had survival data of the cohabitant and shedderfish. And based on these data alone, we could estimate genotype effects on the susceptibility of the cohabitant fish and the infectivity of the shedderfish. So using Bayesian methods, so this shows what you see here. On the two graphs is you see posterior distributions for the estimated relative risk that the cohabitant fish became infected. And on the left, you see the cohabitant fish that are susceptible, that don't have the good allele, the beneficial R allele. And on the right, you see cohabitants that have that allele. And you can see easily there's a huge difference between left and right. So obviously cohabitant fish that have the beneficial allele have a much lower estimated risk of becoming infected. So in fact, the differences were tenfold that was conferred by this one allele. So that resistance allele, which is really defined on whether fish, when they're exposed to the infection, die or not, really does confer also differences in their susceptibility. The fish that have this good allele are less likely to become infected. Now, in each graph, you see the three color distributions. And this gives us information about the infectivity differences between the shedderfish. And again, you see actually there is a lot of overlap. Remember, this experiment was really small scale, but there is differences between these distributions. And again, you see that cohabitant fish that were exposed to shedderfish that carried the beneficial alleles had a lower relative risk than cohabitant fish that were exposed to the shedderfish that didn't carry the resistance allele. So this is why I call this the IPN lucky case, because actually we didn't know a priori what selection for this allele does to the transmission of the virus, but actually it's really good because it reduces the risk of becoming infected, and once you're infected, it reduces the risk of spreading the infection, of transmitting the infection. And I think this this good combination, this positive correlation between susceptibility and infectivity is probably part of the reason why selection for this IPN for that allele has been so beneficial and so good. Now, of course, the question arises, is that the rule or the exemption? So do we always have a good positive correlation between susceptibility and infectivity? So we looked at a different virus. It's the infectious salmon anemia virus, which also infects Atlantic salmon. And this disease is mostly controlled by vaccination at the moment, although the vaccines don't work so well. But also there is genetic selection going on for ISA resistance. But this time we are dealing with polygenic effects. So we are dealing with a disease that is controlled by many genes, and the selection is based on estimated breeding values, again, for survival given exposure. So what we were interested to find out was, does vaccination and genetic selection for ISA resistance reduce the transmission of the virus? And this is important because this disease is a notifiable disease, so you really need to reduce the spread of the infection, not just get animals, fish that cope better with the infection. You need to reduce the prevalence of the virus. And again, we designed a relatively small-scale transmission experiment, which is now a little bit more complicated or complex than the IPN experiment, because we are dealing, first of all, with polygenic effects. So we had to have different families with different breeding values. And we also wanted to see the vaccine effects. But in essence, the experiment had a similar principle as the previous one. We split up fish into shedder fish and contact fish. We needed to make sure that they're equally distributed so that the different families with different breeding values are equally represented. And the shedder fish were either of low or high breeding value, non-vaccinated, or they differed in their vaccination status. So the shedder fish were inoculated, put together in contact with contact fish that were all susceptible, and then we just monitored the survival of the contact fish. So we monitored only the data we had was the time of the death of the contact fish and a confirmation from tissue samples whether they indeed died because of the virus or not. And from the shedder fish, we view the infection status at different time points. And what we found in this case, so this graph here shows now posterior distributions for susceptibility on the left and infectivity on the right. And the different colors refer to fish with different breeding values for resistance. And the first thing you notice, again, the distributions overlap. We are dealing with a small-scale experiment. Yet we see that the overlap between the distributions in susceptibility is much bigger than the overlap for distribution of the distributions for infectivity. So in other words, genetic selection for ISA resistance, these results would suggest that they don't make the fish, they decrease the susceptibility of the fish to become infected, but they decreases the ability of fish to transmit the infection once they're infected. So selection for ISA resistance reduces the disease spread. And the other thing I wanted to point out here is that the blue curve here in the infectivity distribution refers to the vaccinated individual, whereas the green and the red curves show individuals with high and low breeding value for resistance. So I found this very interesting because often vaccination is considered as the first arm of disease control, but actually the effects of the genetics here in this case seem to be bigger than the vaccine effects. So it shows again, we need to really take every tool in the toolbox. Genetic selection can potentially have bigger effects on disease control than other methods such as vaccines when they're not very effective. But note of caution, realize that this is a small-scale experiment, and of course validation needs to be done before this is really implemented. Now, as I said, in the end, we don't want to, experiments are good and we learn a lot from every experiment, we learn a lot, but eventually we want to apply this, to field data. And where we are venturing at the moment is a disease called bovine tuberculosis. It's one of the most persistent animal and also human health problems in the world. So it's a zoonotic disease, which is endemic in many countries. In the UK, where I live at the moment, they try desperately to get rid of the disease. So they have a huge scale eradication program because it causes massive financial losses and there's no safe vaccine on the market at the moment. Now, the good thing about this huge eradication program is that it generates very good disease data. So what they have is they have routine herd testing. So they test every, they have routine testing of herds every year. But when they find an animal that tests positive, they then test all animals in the herds repeatedly every 60 days until no more animal tests positive for 2 consecutive tests. So we have very good disease data. My colleagues in Roslin and also in the Scottish Rural College, they had the wisdom to combine this very good health data with the existing genetic data that breeding companies or breeders are using for selecting cows anyway for milk production. So there is good genomic and pedigree data available for over millions of cattle and very good disease phenotypes, again, from a lot of cattle that lend themselves to genetic analysis. And indeed, there is very strong evidence that there is genetic variation in bovine TB resistance. So it has relatively high heritability. Again, it's polygenically controlled and the prediction accuracies are good enough to implement into genetic selection. And indeed, about 5 years ago, the UK has now launched a genetic selection scheme. It's called TB Advantage that allows farmers to select bulls that have a higher breeding value, a lower genetic risk for testing positive for bovine TB. And this has been, the uptake in the UK has been remarkably good. So it has been really welcomed by farmers. The problem is, as we know, genetics, especially in cattle, cattle which have a low generation interval, the differences, the change is slow. So the danger is that farmers will stop doing it because they don't see fast enough progress. Now, and this is again where infectivity comes in. Can we do better? You know, is it possible? Can we accelerate this progress of reducing bovine TB by also selecting for lowered infectivity? So we looked into that, and indeed there's a lot of experimental evidence that there's huge variation in shedding rates. And when you look in the bovine TB literature, there's a lot of talk about these so-called super spreaders again. So you have phenotypic super spreaders, supposedly some animals that shed a lot. And we did some preliminary quantitative genetic analysis that indicates that the heritability estimates for infectivity may be just as high as for infectivity. So before we venture into the real analysis of the data, we wanted to do a little modeling studies to say, well, what would we gain by also including infectivity in our genomic selection programs? And this is so we developed a genetic epidemiological model. And what you see here is in this graph, we see the relative risk that a herd experiences a bovine TB outbreak over successive generations. So we standardized it that it's at the moment is 1, and then we can see, so if you selected for susceptibility, how would that risk reduce? And if you select also, if you also select for infectivity, how much more, how much faster would you get the reduction? And what you see here is that partly depends on the correlation between susceptibility and infectivity, on the genetic correlation. But generally, you could accelerate the reduction in the genetic risk by a factor of 2 roughly if you also included infectivity into the selection program. So it seems to be worth it because you could speed up the whole process by a factor of 2. So we currently work on reanalyzing the data to get breeding values for infectivity for all the cows that have breeding values for susceptibility. And our hope is that this will eventually lead to TB advantage plus, where we not only select for resistance, but also lower infectivity if we get good results, and then speed up the eradication of the disease in the UK. So with that, I just want to conclude with my vision forward. So in the livestock field, breeding for disease resistance has been long advocated with the genomic tools now, and especially if you listen to José's talk before, really getting deep genomic data and including also functional genomics, we can probably make a lot of progress there. What I'm advocating is we can do even better. We can not only breed for higher resistance, but also lower transmission. And that together will lead to a much more effective disease control than we currently anticipate. And with that, that leaves me to, first of all, thank you for listening and acknowledge all my collaborators. So we have different parts for the method development. We have, oops. Can you see me? Okay, my screen disappeared. So that's just, thank you very much. I don't see the names now, but there were loads of collaborators included in that. And yeah, thanks very much for listening.
Thank you very much, Andrea, for this talk. Really so enlightening and very interesting in the topics. But unfortunately, we ran out of time. So we have some questions. I will ask you to answer them quickly, because another event is coming in 10 minutes in this room. So there is a question for Dr. Casanova. It says, given the results, what are the results of treatment with the type 1 IFN in the clinical setting? Pardon?
Yeah.
And
well,
are these strategies to decrease the antibodies?
So the clinical trials that have been published, most of them of type 1 interferon, whether beta or alpha 2, were in patients admitted, hospitalized with hypoxemic pneumonia, and it did not work, which makes perfect sense because it's too late. There are trials of alpha or beta interferon in ambulatory patients early in the course of infection. And these trials should be published in the coming months. So everybody's waiting.
Okay, thanks. There is another question for Carolina. It says, should Mendelian randomization studies be required when pertinent before considering random clinical trials?
Yeah, thanks for the question. I wouldn't say they should, but it's a good practice to have all the evidence you got before embarking in a very expensive study like a clinical trial. So if the elements are there, if there are genetic instruments available for the exposure that you want to test, then I would recommend to do it, because there's public data sets available that you can try that with minimal cost, and then add that to the observational evidence before doing a clinical trial.
Okay. Well, for instance, we need to end here, but let me say first, really appreciate the talks, because they give 4 different visions or fields that they are developing, and really are interesting, and maybe we can combine these know-hows, because, for example, for Andrea, really is interested in the modeling that you are doing with the spreaders. And I was thinking if we can apply it for humans. Also, for Mendelian randomization, if we need to use another approach, just not only the SNPs, and think in rare variants that Dr. Casanova was talking about in the models, you know, I think that it's a field that is going to develop more, and well, I'm really happy to have you in this symposium.
Thank you.
Thank you, you all.
Thank you,
Thank you.
Bernardo, for inviting us.
Yeah.
And thank you for
Bye.
listening. Bye-bye.
Bye. Thank you.
Bye. Bye-bye.